And of course, we need to thank very much Martha, who accepted to present this webinar to us. Martha is a member of our science board. But of course, the most important thing is that she's, as you know, one of the top phage scientists in the world. And I think we will be really happy and very interesting to hear about her insight about phage ecology. And particularly phage ecology informing applied development, which we are also interested in. So Martha, thank you very much. And we're very eager to listen to what you have to say and of course discuss.
Okay, great. Well, thank you very much for my kind of invitation. It's a great pleasure to give a talk to this audience, this group doing very interesting work in this area. And my talk, therefore, it's a new talk. It's some theories that I've been thinking about for a while, but finally published last year. And then there's also some brand new data which we've managed to capture in the last year, despite everything that was happening. So I hope it fits together okay for your interests. I wanted to start off by explaining a little bit to you why I think it's so important to consider ecology and different aspects of phage biology. And then I'll give some tangible examples from recent work. So the figure on my very opening slide, you will come to know quite well over the course of my presentation to you. Essentially, I'm going to make an analogy to why we need to understand a lot about phage ecology by showing you why it's so important in plants, and how I want to bring to your attention the way conceptually we understand plants and how that can help us understand many useful processes, and why I think we're missing a trick if we don't do the same to phages within the context of disease. So I'll make some analogies to this as we go along. Right. So essentially, there are four things that my talk will highlight in terms of why I think understanding the relationship between phages and their bacteria in the context of us as humans, and the context of where they live in terms of their physical environment when they're not inside us. I'll talk about why understanding these relationships is important, both to understanding the behavior of phages in the system, because if we're interested, for example, in asthma and phages, we want to know what phages are doing naturally within the lung environment, so that we know how to then intervene and add specific phages for specific pathogens. I'll talk about how this understanding can impact the selection of phages and how we isolate them. And just touch very briefly, that'll form most of my talk. I want to just briefly mention something about how it can also influence dosage and timing of application as well. So why do we need ecology and ecological types? Why am I talking about ecology to people of your background? Well, if you think about it in plants, understanding ecology and ecological types of plants really helps us understand fragile ecosystems. It helps us to manage them. It helps us to understand specific environments and how to protect them from invasive forces like farming, and also from climate change. So what? And this is there's a key textbook which I read as an undergraduate and absolutely loved, was by Grime. He was an academic based in Sheffield, and he wrote this book called Comparative Plant Ecology. And essentially what he puts forward in this book is, if you understand the species traits of plants, then you can understand sort of where they fit in terms of like what environments you'll find them in and what their functional ecological role will be, and how you can build communities. But it's all about understanding how individuals actually are. And he proposed these ideas in about the sort of late 60s, 70s, and he spent 30 years justifying them, but people are still using them today to understand specific botanic or plant-based environments. In phages, I think it would be useful for many reasons. So we know in phage biology, I'm sure most of you know, there's been a major overhaul in taxonomy recently, led by folks such as Eugene Koonin, who are proposing we classify phages as according to, not according to their morphology, but according to their phylogeny, and the names have all been overhauled and family levels have been moved up. But actually there's been, and arguably there's some sense in doing that, although it makes it quite confusing, but also, but there's been no sort of there's been no development really in terms of understanding ecology of phages and what they're doing. And that's really what we want to do. As I said in my first slide, we want to try to work out what phages are doing where, why, and how. And therefore this could help us find specific phages for specific bacteria, where sometimes many of the bacteria we're interested in, for example, that are associated with asthma, it's quite difficult to find phages for. So insights could help us direct us to phages in the first place, and then work out how would be the best to use them. Because commonly it's almost as if for every group of phages we're interested in, we reinvent a wheel. You know, we isolate loads of phages and we do lots of tests, and we look at host range and we look at stability and things, but there could be a set of common parameters that we could use to help us get more quickly to useful phages. So let me just talk about Grimes's triangle now. So the way that he classifies the individual traits of plants are according to 3 axes. So these may or may not work for phages, but I thought I'd give it a go. It's worked for 30 years or more, 40 years. People have found it's worked as a very useful framework for phages. Now, for plants. And now, if it doesn't work for phages, we could change these axes. But I thought we'd start with something that's there. So he defines plants as to being some plants have very good competitors. So they what they do is they make a lot of roots and a lot of leaf and a lot of defense systems. So these are things like stinging nettles. Okay, I've tried to use plants that we all know, even non-botanists. So stinging nettles, they have a lot of roots, a deep root system, and these just horrible stinging on the leaves to defend them against animals, stop them being eaten. And eventually, when all that's established, then they flower and go through their cycle. Now, other plants are known as very good stress tolerators. So these are things, for example, wild thyme, or anywhere where you have a stressed environment. For example, chalk grasslands in England, where you have water draining through. There's a water shortage all the time. So the plants are small. They just quickly they have small flowers and they have very small leaves. They're not wasteful with resources. Okay, they can tolerate those stressful conditions. Now, the third axis on his the way he classifies plants is according to this is a slightly more difficult concept in a way. It's to do with disturbance. So some plants are very good at coping with just their environment changing, like fire or flooding. So something like that would be a poppy. So poppies are always seen as being the plants associated with war, because they're the first colonizers on trenches when the ground has been really disturbed. So they can get in and get out quite quickly. So that's their strategy. So you can classify all plants according to these 3 axes, and many plants, of course, are in the middle. Now, with phages, let's now apply the same principle, at least for phages. So potentially, you could have a competitive phage, would be one where the bacterial conditions are good, but what the phage would then do would be to tune the resources in that bacteria to its own needs. So a bit like that stinging nettle. So it'll essentially make its version, it'll make an infrastructure. So it'll change the transcription earlier on of the bacteria so that it can do the things that phage needs. A stress phage, in comparison, would be one that can cope with a stressful environment. So something like a cyanobacteria in the oceans, the cyanobacteria are very stressed. The phage have to live in that high light, low nutrient environment. So a phage would have genes to allow the host to adapt to that. And a R phage would be like a quick in and out phage. Okay, so that's the theory. So how this might work. Now, how do you tell what phage, how on earth do you tell what strategy a phage would follow? So this is me with the boys a couple of years ago in a chalk grassland, and my dad made us sit still and just see how many species of plants we could identify in our immediate arm's length. We could identify 30 different species, because nothing can dominate here because it's so stressed. You've just got a really nice diversification of stress tolerant plants. The picture next door, these are my Clostridium difficile hunting grounds for phages. So we could potentially find lots of phages there, but there's no such textbook that exists, so we haven't got the scheme up and running. So how would we do this? Well, phages would have life history or biological traits, which is to say that they that any trait that allows them to adapt to the environment that we find them in. So a biological trait could be used to sort of understand these underlying mechanisms, and we would expect that to respond to different gradients within an environment. So what kind of things, what species traits or biological traits could we measure in phages? Well, we could measure things like morphology, specific behaviors of phages. We can look at temperate, lytic, we can look at genomes, tRNAs, polymerases. And one of the problems we have is that, unlike the plant world, because phages are relatively diverse, it's difficult to find something that's a good common trait that's that the majority of phages will share. So this has been this is challenging. But one thing that they all do, we know that when they infect their hosts, in general, they have 3 phases of transcriptional takeover. Okay, so I thought to myself, I wonder if you could profile them and understand them in terms of how many genes they switch, that they use to direct transcription to their own machinery. So how complicated is their redirection of the bacterial metabolism? So you'd think that would be the early genes that the phages have that do that. So that could be, so that's a little bit like a competitive, that would be like a competitive strategy. Or how many genes are the phages expressing in the middle stages? So that would be a sort of stress response. And finally, how many are they just immediately going to their final morphogenesis? So how many genes are associated with that? So that's what I just thought. Well, I wonder, actually, this could be a really, really neat fit to the plant framework to see if the idea worked, because I like this because it's not subjective. Many of my other ideas about immediately measuring phage behaviors possibly are more subjective. But if you look at the transcriptional profiling of phages through RNA-seq analysis or through other techniques, we can see very robustly what genes are expressed where and when. So let me just show you how this looks in different phages. So we know that phages, after they infect their bacterial cells, they either infect, they have these 3 profiles, as I said, early, middle, and late. So these are genes that allow them to interact with the bacteria, replicate, and then go into morphogenesis. So this is what, let me just show you here. This is where such phages would be plotted on that triangle. So here we have a Streptococcus phage. So that's sort of in the middle of our triangle because it's got a fairly equal number of genes involved in each process, compared to a Prochlorococcus phage. So this is a cyanobacteria. It's got loads of genes involved in stress. So this is a stress phage. And this final phage I'm showing you is T5, an Ecology phage, which has just got 2 profiles. It expresses things early and late. So this phage is up here as a competitor phage. It immediately changes transcription to suit its own purposes straight away. So this is the idea which we thought we'd try. Now there are 42 phages in existence that have had their transcriptional profiles done. So not very many, considering there's about 9,000 phages in isolation. And the first plot I'm just showing you now is just, it's just all we did was take all of the 42 phages that have been looked at and look to see what the early, middle, and late genes are. Does this roughly correspond to what they should do? So do early genes correspond to do early genes correspond to the transcriptional takeover, middle stress? We can see that they do indeed. There's lots of stress genes, such as those involved in phosphate starvation and different things a bacteria might need when it's under stress conditions. And the late ones are indeed responsible for morphogenesis. So it looks, if you take the genomes, the annotations of genomes that have had their phage transcriptional profiles looked at, this looks like it fits with these ideas. And indeed, this is now just to show you where the 42 phages are on this triangle. So we can see, I've just labeled 3, I've coded 3 groups. And if we look, we can see the all of the cyanobacterial genes that, phages, they're all green. And they're all phages associated with these living in these stressful environments. The Pseudomonas has had quite a few phages looked at in terms of their expression profiles. That's mainly been done by Rob Levine's group, and some of these are indeed phages which are useful therapeutically. And you can see they're clustering in different groups. They're in quite a lot of them are in the middle group, the CSR, and then the clustering. There's a large phage, Phage, that's clustering as a competitive phage. And then they're also spanning these other 2 intermediate strategy groups as well. And E. coli are also quite spread out. Now, in general, many of the phages that have been looked at from a transcriptional point of view not very much is known about their biology. So I can't say a huge amount, but I'll just pick out one phage from each group just to show you it does seem to make biological sense at some stage, which is nice. So with the C phages, we found 7 phages that were in that group, and they're all infecting bacteria that, at least in some periods, we know will be there'll be a nice surplus of bacteria for those phages to be able to infect. And let me just pick out this phage T5, which I mentioned. So we know that T5 is immediately after infection, it inhibits RNA synthesis and protein synthesis of the cell, and it degrades host DNA and excretes things from the cell. So we know that it's a phage that does a lot of things to early metabolism. So that makes sense, it's there. S phages include T4, the E. coli phage, which is a big bulky genome full of many genes that manipulate the metabolism of E. coli. And we know, for example, that T4 can survive infection when the cells are starved of oxygen. I'll come back to oxygen and the impact it may have on phage infection, particularly in an asthma setting later. So there's plenty of these stress phages that we identified. We know that CS, so the T4 is a sort of borderline CSR, but we know that it has things, for example, it encodes a lot of its own nucleotide metabolism. It replaces the bacterial metabolism with its own version of it for its, as it goes through its phage life cycle. Now, in terms of the R phages, we've got a few phages were identified in this category. These are your quick in and outs. The one that most is known about is a phage called Pha, a bacillus phage. And we know that it's got quite a complicated set of sigma factors and structural proteins that the phage can regulate. So this was this started off as an idea to see if this may work. And I think hopefully I've shown you some evidence that looks like it. By thinking of phages in terms of functional types, we can therefore start to understand their biology a little bit better, and then we can say things like, for example, what we want to know is, so the phages that we want to use therapeutically, do they will they be more suitable to, are they found in particular categories? So if you want to use a cocktail, for example, of phages to treat Pseudomonas, are they going to be are they C strategists, or is it best to have a C, an S, and an R, or where perhaps if we're just selecting phages because we know that we can bulk them up and make them up really efficiently, maybe we're actually selecting for R phages, where actually we want phages that can cope with the stressful conditions found in infection. So we may be after S phages, if you see what I mean. If hopefully by trying to understand what strategies may correlate to the features that we want, I think it would allow us to put together stronger, more informed cocktails of those phages. So if anybody is working on specific phages and is doing transcriptional analysis, we've made a free little program for people to access and put their phages in to see where they fit on this triangle. So that's a tool that myself and Thomas Sikkerhans has developed. He's based in University of Copenhagen in Denmark. Right, so now what I'd like to do is relate what I just told you to how this might be useful and interesting in a lung phage context. Okay, so we know that phages in the lungs, like anywhere else, but we're interested in the lungs at the moment. We know that different having a high diversity of phages or a low diversity of phages may be related to disease. We also know that the interactions between phages and hosts can result in having, and this is in my number 2 in my little schematic, can result in changing of the phenotype of the bacteria. So for example, it can become a lysogen and get better at making biofilms or invading epithelial cells or producing toxins, or alternatively, to escape phage predation, it may change its outer surface again, which may give it different properties. Or it may have a phage just infecting in a non-lysogenic way, but existing as a circular plasmid, again conferring extra properties to that bacteria. And also different conditions with different physical environments can select for lysogeny or not lysogeny. So this could impact the dynamics of between and the impact of the bacteria on the human host. And finally, phages themselves directly may have a direct impact on disease. So we know this, but we don't really have any understanding at the moment about what types of phages may be involved in these different sets of relationships. So what I'm showing you now is some data from Chris Breitling, who's based at University of Leicester. Now he works, I'm sure some of you may know him, he works a lot on COPD, and he's shown, I mean, to me these numbers look, they're kind of small differences, but this is published data, and essentially he's shown that if you look at Streptococcus haemophilus and Varicella in COPD, you can see that during exacerbation you have slightly lower numbers of, in general, this is from a particular patient cohort, you have slightly lower numbers of Streptococcus and higher numbers of Haemophilus. But it's very difficult to see in this, and presumably with asthma as well, what the difference is. So when you've got bacterial just colonization, and when those bacteria are actually causing infection, and what's causing the flare-ups and the exacerbations. So these dynamics can be quite hard to understand. So really, I guess what I'm trying to, what I want to bring in is that perhaps when we understand particular phage types, then we can see, oh yeah, we've got these, we'll be able to link phage types to different disease states. Now, I just wanted to sort of mention a couple of different examples of differences in physical environments that you might see, for example, within a lung, that could change very much the dynamics and whether or not you've got disease. So this is the concept of trying to understand what phages are doing naturally. So the first data, this is actually quite old data, but I just wanted to mention it quickly because we have made some progress on understanding it further. So we noticed some years ago that when we looked at the respiratory pathogen, Burkholderia pseudomallei, the phages that we mainly found after a large screen in Thailand, where this is a particular problem, we noticed that when we infected at 37 degrees, we could kill the bacteria. So that's the top bar in the chart. And then when we infected at lower degree, at the bar below that, the dark gray, when we infected at 25 degrees, the phages didn't kill. And we did quite a lot of work, and we essentially showed that the phages changed their lifestyle according to temperature. So at high temperature, they go through the lytic life cycle, and at a low temperature, they go through the lysogenic life cycle. They become lysogens. Now, we did a lot of further work now to show whether the temperature of the switch is actually at 31 degrees. So you can easily have a situation in the lungs where there's sort of, there could be minor differences in temperatures could have quite big, profound impacts on the relationships of phages, and this is just to show you visually that when we can pick out lysogens of this phage, and we can see that when we grow them at cool temperatures, bacteria grow, and at higher temperatures, the bacteria pop out. And we think we've done work, we've made temperature sensitive mutants, and we think it's a riboswitch in the phage that's controlling this behavior. So I only just wanted really to touch on this briefly to say that I think you could imagine that perhaps particular types of ecological types of phages would be associated with the ability to work at different, optimally under different environments. So the next thing I want to tell you about is related to oxygen status. Now, the reason I wanted to tell you about this is because it struck me when I was putting my thoughts together to make a seminar for you, that although you're thinking about a lung environment, within that lung environment, you've got different microenvironments that would have more or less oxygen. So you can imagine if you've got a bacteria growing as a biofilm, underneath that biofilm, there could be no oxygen or much less oxygen. Now, this is an area that people have really not explored very much in terms of phage bacterial relationships, but hopefully I will show you some data that shows you why I really think you should, because it could be very profound in terms of how those bacteriophages are infecting the bacteria. So this is relevant both to understanding what's going on and also to applying phages. And I think that by not taking this into account, we may use the wrong phages. If we find a set of phages that work really, really well under full oxygen conditions, they may not work in the conditions that we want them to work in. And also just by when we're pulling apart these systems, if you don't take samples that are truly reflective both of the area and also the time that you're interested in, when you have a difference of disease severity, you may miss what's going on, and certainly at the moment we have no idea what types of phages may be responding differently to the different microenvironmental pressures. But let me just convince you now that it is that there is a difference in terms of oxygen status. So here we have a few phages that were isolated by my student Francesca Hodges, who's a PhD student in my lab at the moment. And this is the host here. This is actually this is an Aeromonas host. Can you see here this is infecting this little podovirus, and it infects differently. These are the plaques you can just about see if you squint anaerobically, whereas aerobically it's making nice big plaques. In contrast, this is a Proteus mirabilis bacteria as a host, and we can see these are the phages infecting anaerobically compared to aerobically. So we can see there are many different. She isolated 19 different phages actually from different types of hosts that are associated with shoulders. Actually, it was a completely different project, but the principle is there, because these are bacteria people haven't really studied very much before. And this type of work, as I say, is barely anything in the literature. We just made a review, actually, it should be coming out soon on this topic. But what you can see here, now this is, these are a whole bunch of phages, 6 phages that infect one particular bacterial host. And I just want you to see that this is what's known as the efficiency of plating. So it's showing how efficient they are at being able to plaque under the different conditions. So blue is anaerobic and red is aerobic. So we can see some phages work better without oxygen, and some work less well with oxygen. And some of them there's a really big difference. And some of them, look at phage 3, really does not like oxygen. And then this is another host, and again we see similar things. Some phages are much better at infecting in the presence of oxygen than others are, and some clearly better anaerobically. Now, of all the, I just want to show you, I don't need to go into the detail of this here, but just this is now what's known as, we call it a one-step growth curve. All the phage biologists have done these millions of times, but essentially, can you see there's quite different dynamics, whether we look aerobically or anaerobically, in terms of the number of phages that come out and the time take from when they start to be released from the cell following that latent period. Now, this is just showing now, this is some of the data summarized. We can see that the things that we're interested in when we use phages therapeutically, we care about burst size. And we can see that the burst sizes are quite different in many phages between those two conditions. And the latent periods can also be quite different as well. And how efficient they are at infecting also differs. And we actually had, we studied 19 phages that infected several different hosts that came from the same source, which is shoulders. And we can see basically 9 of them infected better under aerobic and 10 under anaerobic conditions. So I think going forward with that, it'll be interesting, as I say, both to try to understand better what might be happening in terms of, so when you pull out model systems to try to figure out what they're doing, I think this should be, looking at phages with this way could be quite insightful. Right, so the next little thing I want to mention, hopefully again will be of interest for this work. It's just, so I wanted to think about where we get our phages from, and how sort of being, I guess, ecologically informed may help us get phages from sources that are likely to work. So very often a common source of phages is to get them from sewage. It's an easy source, and there's many bacteria. But I think we're probably only seeing a subset of phages. So I have some rather exciting projects at the moment going on, where we're looking very broadly at species-rich environments. There's a project that I'm working with folks from Denmark, Malaysia, and Colombia to look at sort of very species-rich environments. So after showing you lots of data and graphs, here's just a picture of a nice, this is inside a tropical rainforest in Malaysia, one of the oldest rainforests in the world, where we're looking just to see what types of phages we can pull out from very, very species-rich, more or less untouched environments. So both we're looking for phages that target pathogens that we know, and looking at diversity in general. So these are some of our top, these are much more closer to home environments that most of us sample in, so sewage works. But even just going actually not so far, not so exotic, but just looking at this top figure here is looking at a massive horse muck heap near where I live. And we found some new, very new and interesting Pseudomonas phages in those environments. Now, this is looking at, so C. diff is an anaerobic bacterium. So I found, after lots of unsuccessful sampling of ill patients, I went to the salt marshes and we found good phages there. And again, this is actually a project on Borrelia looking at Lyme disease. So the best place to look for such phages. The Lyme disease is carried by ticks. So we're looking in environments associated with ticks for those diseases. So it's taken me, probably most people know this already, but I feel like it's taken me quite a long time to sort of figure this out. And I think it's, in that if you want to find phages to treat people, you're not, it doesn't seem to be you get very good, you don't often know very good phages associated with those ill people, but perhaps going to healthy people, where phages are naturally playing probably more of a role than we think, can be a really good source. And the examples I wanted just to mention that I found that to be useful recently, one was with a disease called Rhodococcus equi. It is a very, very, very nasty pathogen, respiratory pathogen, but for horses. But if a racehorse gets it as a foal, it will never race. So the racing industry really care about this disease. It causes them a lot of wastage, and it's very sad for the poor horses. Now, there were no phages at all that were known before I started working on it that could actually infect at 37 degrees, which is the temperature for a horse lung that we'd want them to be able to infect at. So I actually just went with my student to lots of really healthy stud farms near where we live in around the place in Leicestershire. And we found from that, we found phages, because there were some Rhodococcus phages, equiphages, but they only infected at 30. So we actually used the conditions we want them to be able to infect at 37. So we isolated at 37. And this little schematic here, it's showing a cloud, showing the clusters of relationships of all the Rhodococcus equiphages. And I just want you to look at this little cluster sort of to the left side that's got 4 purple phages. These are our 4 new phages. They have some relationship to phages that are known about, but their genomes are quite different. And they infect really, really well. So presumably in natural, healthy race horses that aren't getting Rhodococcus equi, they have these phages that are playing a kind of a role. And this was the same. Also, I did a project recently looking at salmonella in pigs, in UK pigs, and I collected pig fecal samples from healthy pig farms around the place, and we found one really quite a distinct family of salmonella phages that hadn't been looked at before. I think I've highlighted it here. Yeah, this is showing a cluster analysis of all salmonella phages. So this is just my cluster from my new phages. And it was quite interesting because those farms are nowhere near each other, but I kept finding this variance, very similar variants actually, of this one phage. And they're big phages, they're about 230 kilobase genomes, big phages. Probably, we haven't done the transcriptional profiling, but I predict they're going to be S phages because they're big and bulky and they do an awful lot of things, probably CS phages. So we found this new family of phages, and we can see here, this is more cluster analysis, just showing that they're quite distinct from anything that's been known about. This is relating it to other salmonella phages on the left, and on the right, I'm relating it to all the other giant phages and other species, because there's some similarities when you look at these, what we call these jumbo phages over 200 kb. Now, what was interesting is just how effective these phages were. So in the UK, salmonella, there's 1, 2, 3, you can see these 5 major serovars of salmonella. And my phage they were brilliant. They could just infect nearly everything. So they're very, very good broad host range phages. And we've just finished actually a trial of, we did because the strains that infect chickens are very similar. We did a chicken trial, and the phages worked brilliantly. They completely eradicated the carriage in a challenge model. We had a trial with 700 chickens. Okay, so that just sort of leads me to my last point, which I didn't want to. You could give a whole talk or think about this for a whole other talk, but I just wanted to end with thinking about when do we use phages, and what about the dosage and the timing and the combinations of phages. Again, I think could probably really benefit from understanding, does this make a difference if we have different phages of different types and different strategies? Can we figure out some of these aspects of deploying phages as well? So in the recent case of my that I just did this trial, I mentioned the salmonella phages. I was quite interested. Nina won't be at all surprised by this, or probably not Jean-Paul either. But I'm not sure I was surprised, but I was really delighted because it was a big study. As I said, 700 birds, and we saw that we gave the birds salmonella at day 4. They had phages embedded from their feed all the way along. So they what we wanted to see is what the dosage of phages we should add. Did it make a difference? So we chose the dose. We knew that how many bacteria there would be per gram of intestinal material in the chickens, and we did a 0.1 ratio of that to phages, 1-to-1 ratio, and a 10-to-1 ratio of phages to bacteria. And what we can see, if you just look here at day 42, we can see that the lowest dosage of phages, so 0.1 dose of phages to bacteria, completely eliminated the salmonella from the chickens. And this is really interesting though. So there's a clear difference even in a 1-to-1 and a 10-to-1, but the best results were seen from this lower dose. So yeah. So that's just sort of wanted to finish with that observation. But I think to sort of try to bring what I said to a conclusion. Hopefully I've sort of shown you, I think, that by looking at phages in terms of their strategies and behaviors, it could really transform our understanding of the way they work. So for example, I was thinking that, in the bacterial world, we understand that some bacteria, for example, are opportunistic pathogens. We know that, things like E. coli and Pseudomonas. We know that other things are, like Streptococcus, are very host adapted. We know that certain bacteria are always intracellular. We understand them. We know how to think about them and get rid of them if we need to. But we just don't have this yet for phages. So I think that we're sort of missing out by not having this understanding. And I don't know yet if the CSR will be exactly the right framework, but it looks quite promising so far. So with that, I will finish, and hopefully I've left some time to have a nice discussion on these topics. And thank you very much for your attention.